Afternoon, everyone, and welcome back. I would like to present to you Kavya and Christi, who are going to do a presentation on their summer research, which is on the effects of exercise and doxorubicin on cardiotoxicity. So welcome, Kavya and Christi.
Okay, so hi, I'm Christi Cook.
And I'm Kavya. And the project that we've been investigating is the effects of exercise on doxorubicin-induced cardiotoxicity on circuit 2 expression. So the first thing to talk about is our drug, which is called doxorubicin. So doxorubicin is a drug, it's an anthracycline, which is a drug that's derived from the bacteria called Streptomyces. And this drug is very, very useful for chemotherapy treatments because of its ability to damage DNA. And there's two main ways that it hurts the DNA, and the first one is through intercalation, and the second way is through inhibition of topoisomerase 2. So in the first picture, we can see this is an example of how topoisomerase 2 works. So this protein complex basically helps the DNA unwind so that it can replicate, because when it's in its helix structure, it can't replicate. So it helps to unwind. But when doxorubicin is present, it inhibits the function of topoisomerase, and then that hurts the DNA, and then that means that replication can't be successfully completed, and as a result, cell apoptosis happens. The second way doxorubicin hurts the DNA is through intercalation, and we can see this in the middle picture right here. Basically what the doxorubicin does is it inserts itself in between the two strands of DNA, in between the nucleotide base pairs, and that the DNA strands zip around it, and then it can't replicate. So this is the two main ways that doxorubicin damages the DNA and causes cell death. And cell death is obviously really good for the cancer cells, but it's really bad for the heart cells, which the doxorubicin unfortunately reaches really easily because the heart has such few protective enzymes. So that's why doxorubicin has been claimed as a very cardiotoxic drug, which means it hurts the heart. And so not just through DNA damage and cell death is doxorubicin hurting the heart, it also is involved with production of free radicals, which therefore causes oxidative stress. And this happens all in the mitochondria. And basically, there's a protein in the mitochondria that helps to maintain the iron levels, and what doxorubicin does is it inhibits the function of this protein exporter. So what ends up happening is that all the iron accumulates in the mitochondria, and the doxorubicin reacts with it to create a bunch of free radicals, which end up causing oxidative stress, hurt the mitochondria, and then end up hurting the heart function. Okay. So the main mechanism that we decided to look at in terms of doxorubicin's cardiotoxic effects is a pump called circuit 2, which stands for sarcoendoplasmic reticulum calcium HPase pump. Basically what this pump does is it regulates the intracellular calcium levels, and this is a really good way to measure the heart functionality because many studies have been done where the gene that codes for circuit 2 has been knocked out, and what ends up happening is that the heart's ability to contract and relax decrease a lot. So that's why this is a good mechanism to study. And basically how circuit 2 works is it's regulated by a protein called phospholamban, which is this purple one right here. And so when phospholamban is phosphorylated, the calcium ions move down against the concentration gradient from the cytosol to the sarcoplasmic reticulum. But when phospholamban is dephosphorylated, the calcium ions stay where they are, and it just helps to maintain that homeostasis. So that's just an overview of how circuit 2 works.
So in the past studies that we looked at, there was a lot of studies on like how the exercise helps improve the circuit 2 expression, and that, but most of these studies had a longer training protocol or exercise program that ranged anywhere from a minimum of 4 weeks to a couple of months. And so since we figured some cancer patients may not be able to participate in a training program that long, we figured that we should be able to improve their quality of life in a shorter period of time. So the purpose of our study was to determine if the shorter exercise program had the same effects, counteracting effects on doxorubicin as the longer training programs. And we hypothesized that the doxorubicin will negatively impact the SERCA2 expression, but that exercise will help counteract these negative effects.
Okay. So for our methods and materials, the subjects that we used were 16 juvenile frog belly rats, which are very commonly used for laboratory purposes because they're so easy to handle and very good for breeding purposes. And so these 16 rats were split into 4 even groups. The first one being sedentary, which means they did not participate in the exercise protocol with the saline injection. The second group was sedentary with the doxorubicin injection. The third one was exercise with the saline injection, and the last one was exercise with the doxorubicin injection. And this was our exercise protocol. So by short, we mean 2 weeks in our experiment. So in the first week, each day they increased it by about 10 minutes. So Monday was 10 minutes, and they ended up doing about 50 by Friday. And then the second week, they did 60 minutes each day, and they had a rest on the weekends. And they ran at a speed of about 30 meters per minute on the treadmill, which is a running speed.
So in this slide is the isolated perfused heart machine. And so ideally after the rats are sacrificed, you take the heart and you hook it up to this machine, and it perfuses the heart, which essentially cleanses it of the blood. And they also pull functional data from this by measuring the pressure of the heart while it's beating. And so they do this by pumping a Krebs buffer through it, which in the Krebs buffer, there's nutrients that essentially keeps the heart alive long enough to pull the data from the heart. So that's a rat's heart hooked up to the big machine that you can't see.
Okay. And I forgot to mention previously when we were talking about the groups that the control was the sedentary and saline injected treated rats. So with the heart tissues, basically what we did is our job was to extract the proteins, which we later put in a kit, the ELISA kit, which stands for enzyme-linked immunoassay, which we'll talk about later. So to do this, we cut up about 1 gram of the left ventricular tissue, which we can see on the left here. So that's like the heart before it was homogenized. And then in the middle, this is a homogenizer, and this basically helps us to break up all the proteins in that tissue. And then on the right, we can see the after homogenized tissues, which is basically like a fine pink liquid.
Okay, so in this slide is the F-Endorse tube that has the supernatant liquid and the pellet, and the supernatant contains the heart tissue proteins, and the pellet is the leftover heart tissue. And so we used the yellow material here in our ELISA kit, and we filled 44 wells with either 50 microliters of the standards that we were given in the kit, or with our samples. And then from there we filled all of the wells, like even the controls and the empty ones, with 50 microliters of the sample diluent and 100 microliters of the horseradish proxidate conjugate renegade antibody. And this is like, this HPR was designed to bind with the antibody SERCA2 proteins, so it gives like the accurate, like it brings out those antibodies. And then we incubated it for 60 minutes at 37 degrees Celsius, and after incubation we washed the plate out 4 times with a wash solution, and then dried it and added 50 microliters of chromogen solution A and 50 microliters of chromogen solution B, and then incubated it for another 15 minutes at 37 degrees Celsius. And that turned the wells on our plate blue. And after the incubation time, we put 50 microliters of the stop solution, which is a sulfuric acid that stabilizes the staining process so that the plate reader can identify the proteins and specific antibodies, and that's what turned the plate yellow, which you'll see in the next slide.
Okay. So this is This is a picture of the wells that we filled up with the samples. So as Christi was saying before, the supernatant, which is a light yellow liquid at the top, that's the sample that we placed in all these wells. So on the left is where all our standards were placed, and the standards are reading the concentration of the SORCA2 in each well, and so the darker the blue means that there's a higher concentration. So in columns like 1 and 2 here were our standards, so like each standard was set to be a certain concentration, so that's why they get darker as they go down. And then in columns 3, 4, 5, and 6 are our samples, so they vary in concentration depending on what test group they're in. So with all the data that was shown in the table before, the blue one with all the optical density values, we ran a statistical test called a two-way ANOVA test. ANOVA stands for analysis of variance, and it's two-way because there's two factors, whether you exercise or not and whether you're treated with a drug or not. And so basically what this test helps us do is it helps us to assign a numerical value to each of the factors on how much of an effect they had on SORCA2 expression. So as you can see here, the percent of total variation basically helps us to see how much that factor caused the variation in the sample. So if it caused the variation, that means it has an effect. So we can see here that drug, which is doxorubicin, had the highest percent of total variation, which means it had a statistically significant effect on SORCA2 expression. And this is further proven by its p-value. It's clearly below 0.05, which again means that it had a statistically significant effect. And so activity, so whether or not they exercise didn't have an effect based off the statistics and the interaction of those factors. So combining the drug and exercise didn't have a significance either.
Okay, so this is the optical density adjusted graph. And basically adjusted is like taking into the factors like light and temperature of the standards. And so the linear line right here is ideally lit like it should be. And so our squared number was 0.98, meaning that the standards didn't vary very much from the what ideally they should be, meaning that our standards were like set as a good solid and they ran well.
So basically the only purpose that this oscillator serves is to help us know that the standards were good standards to run and that the data ran well. And our R-squared value, which helps us know how well the data correlated together, since it's 0.98, which is very, very close to 1, that means that there was no major outliers and not a super weird variation. The data was pretty well correlated together, which we can also see by this. Sorry. Okay, so we have another bar graph here, and this is showing the 4 groups and their optical densities. So here we have sedentary, and here's the treadmill, and the black is the saline, and then the gray is the dox. So the main thing that we got from this graph is that we wanted to look at the 2 doxorubicin treated samples, which were this one and this one. And what we were hoping, based on our hypothesis, is that the exercise doxorubicin samples would actually have a higher optical density, which it didn't. We can see that the sedentary doxorubicin sample actually had a higher optical density than the doxorubicin exercise samples. So this helps us to conclude that the exercise had no effect.
So the major outliers for our data, there were no major outliers or major deviations in our standard samples or standard samples. So this shows that our data is really accurate and it doesn't vary much. So, and then also the data showed that doxorubicin has a significant effect on the SORCA2 expression. Exercise protocol was not significant enough to impact the SORCA2 expression, and that the interaction of both factors, the drug doxorubicin and exercise, is also not significant enough to impact the data. And we figured that in future studies, ways to improve this was to have a larger sample size, so you could pull more data. and also to use newer tissues, as we were using ones that had been like frozen for a while.
And also to make sure that maybe next time, like with the tissues, we could actually extract functional data from the isolated perfused working heart machine, because that can definitely give us more evidence to see how well those hearts were working before.
So some of the references are image credits. And then we would like to thank some people for being able to do this research project. Union Pacific Foundation, Excel Energy, University of Northern Colorado, and Math and Science Teaching Institute for allowing us to use their equipment and all their living on their property. Especially Laura Gall and the FSI staff. Thank you for guiding us and putting this whole thing on for us. Stokes and Toast for being there late nights, correcting us, telling us when our flashcards are obnoxiously loud. And also Nicole Wood for being our amazing mentor and advisor, checking all our papers. And Colin Quinn for allowing us to use his tissue from a past study. Also, we'd like to thank our families.
Thank you.
Thank you.